Metabolic pathways comprise only 12% of MPNP. Most other maps and tables deal with biological processes such as genetic information processing here. And you see that, as expected, this is subdivided into replication, transcription, translation, and also some information about chromosome structure, epigenetic, and histone, their modifications. Let's go to replication and to DNA replication. We see here the map with all its components. You see that there are proteins which do not have EC number next to them. In order to get access, we just go to the name of the protein itself. We have here links to purine and pyrimidine metabolism, which provide the substrate for DNA replication. And then we have a table which essentially show all the protein and enzyme which are involved in DNA replication. Here on the right we have a group of links that go to DNA repair of different modes. Now we go to transcription, and you see that this is subdivided into 3 different subcategories because it's a very intricate process. We go to here the biogenesis of RNA, and we can see here the information about the different polymerases. RNA polymerase 2 that transcribes messenger RNA with all the factors and so on, and of course splicing of pre-messenger RNA, which is a rather complex feature here. And lo and behold, there are even some inhibitors that can be found for some component of this pathway. RNAs are extensively modified, and we can see here like tRNA modification, which show the tRNA transfer RNA molecule with all the modification site and all the enzyme that modify these sites. Now we go to another aspect of transcription, that of snRNP assembly. For example, we go to maturation of snRNPs, which require both nuclear and cytoplasmic regulatory step. You see it's quite complex, but it gives you all the available information for this Now we go to translation, and of course the obvious thing to look for is protein biosynthesis. And here we have this integrate map, which contains not only all the amino acid tRNA ligases, but also the elongation and activation factors, and the termination, and the dissociation of the ribosomal unit, and reassociation, and so on. And here we have, for example, signal recognition particle mediated targeting of membrane secretory protein, which leads the SRP together with some elements into the membrane, where it is translocated across the membrane by the SEC We have also found information about transcripts which are translationally regulated in asexual blood stages, and you see here quite a large table which involves many genes that are essentially regulated during translation. Translation in the mitochondrion and in the apicoplast is more akin to translation in prokaryote, and here we have assembled a map which describes all the factors which are involved in this process in the mitochondrion and in the apicoplast, although, as you well know, most of the gene product involved in this process are essentially replicated and transcribed by the parasite nuclear genome.